Hello, welcome to our cell division restaurant project. In our group, we have Nicole, Luca, Karen, and Andrea. Why do cells divide? They divide because the larger a cell becomes, the more the cell uses its DNA. Also, a bigger cell is less efficient in moving nutrients and waste across the cell membrane.
First is the G1 phase. Cells do most of their growing during the G1 phase. In this phase, cells increase in size and synthesize new proteins and organelles. The G in G1 and G2 stands for gap, but the G1 and G2 phases are actually periods of intense growth and activity.
New DNA is synthesized when the chromosomes are being replicated. The cell at the end of the S phase contains twice as much as DNA as it did in the beginning.
G2 phase. When DNA replication is completed, the cell enters the G2 phase. This is usually the shortest of all the phases in interphase. Many of the organelles and molecules are produced. Now the cell is ready to enter the M phase.
Metaphase is the second stage of cell division in which the sister chromatids are pulled apart and moved toward opposite poles of the cell.
In metaphase, paired chromosomes that were duplicated line up across the center of the cell. It's the shortest stage. The spindle fibers connect the centromere of each chromosome to the 2 points of the spindle.
The third phase of mitosis, anaphase, begins when sister chromatids separate and begin to move apart. During anaphase, the chromosomes separate and move along spindle fibers to opposite ends of the cell.
And finally, telophase, the phase of mitosis in which the distinct individual chromosomes begin to spread out into a tangle of chromatin. This is the final stage of mitosis.
Cytokinesis is the part of the cell division process during which the cytoplasm of a single eukaryote cell divides into two daughter cells.
The cell cycle is important because it's the series of events where a cell grows, prepares for division, and divides to form two daughter cells.